Well, good afternoon, everybody, or almost good evening in some parts of the country. My name is Jim Kellner. I'm a pediatrician and an infectious diseases specialist at the University of Calgary and Alberta Children's Hospital in Calgary, and really pleased to be here on behalf of the CanCOVID group, Drs. Vivek Goel and Susan Law, and the CanCOVID Secretariat, who have put together the CanCOVID workshop on vaccines today, talking about COVID-19 vaccines. And we're going to have several presentations this afternoon, and then some discussion, and then an open question and answer session. And so really looking forward to the presentations that are going to be made. And I'm just going to take a minute to introduce briefly each of the speakers that we'll be hearing from, and then outline how we'll conduct the question and answer afterwards. So the speakers in order: first, we're going to hear from Dr. Joanne Langley, who's a pediatrician and infectious disease specialist and professor of pediatrics and community health sciences and epidemiology at the IWK and Dalhousie University in Halifax. And she's also the CHR GSK chair in pediatric vaccinology at Dalhousie University. So Dr. Langley is going to speak on what is the portfolio of vaccines and their key features. And then she'll be followed Dr. Megan Bettle, who is the Director General for COVID-19 Regulatory Response Team at Health Canada, is going to speak on the regulatory process to approve COVID-19 vaccines in Canada. And then she'll be followed by Dr. Carolyn Quach, who is a professor in the Department of Microbiology, Infectious Diseases and Immunology at the University of Montreal, and also the current chair of NACI, the National Advisory Committee on Immunization. And then Dr. Jeff Kwong from the University of Toronto, who is the program leader for populations and public health research programs at ICES, and a scientist with Public Health Ontario, and also the interim director for the Centre for Vaccine Preventable Diseases at the University of Toronto. Dr. Kwong will be speaking on vaccine surveillance in Canada. And after all of our speakers have made their brief presentations, we'll have sort of a summary comments made by Dr. Jim Talbot, who is adjunct professor at the School of Public Health at the University of Alberta, and the former chief medical officer of health for the province of Alberta and territory of Nunavut, and former director of the Alberta Provincial Laboratory. And Dr. Talbot is going to provide some summary comments, and then we're going to open it to questions and discussion. So along the way today, if you have questions, you can put them into the chat box, or you can raise your hand. Depending on how many questions there are and how many people wish to ask questions even verbally, we'll see how much time we have. We may not get through everything, but we'll do our best to address the main questions and have that along the way. We won't pause for questions after each of the speakers. We'll have each of the speakers give their presentations, Dr. Talbot give his comments, and then go to the discussion. So with that, and everybody's participating by Zoom, and just ask that you keep your video off and keep your sound off unless you're speaking, and each of the presenters will have their video and sound on, of course, and so we'll get going with it. So I'm going to turn things over to my friend and colleague, Dr. Joanne Langley from Halifax, to make our first presentation.
Thanks so much, Jim. I'll just share my screen. Good. Good. So you can see that slide? Excellent.
Yes, you can see it fine. Yeah.
Super. Thanks so much. And thank you to the organizers for inviting me. I must say I approach it with trepidation because the level of science literacy, let alone in the academic community about all things COVID, let alone my not my lay friends, is amazing. That we've really advanced in our ability to understand vaccinology over the past year. So I think we'll have a great discussion. So my topic is, what are the characteristics of the COVID-19 vaccines? This is my disclosure statement. Our research center does research with industry as well as investigator-initiated research. And when it's with industry, the funding is paid to Dalhousie University. And my Membership in not-for-profit advisory boards or government committees is listed here. The objectives for this short time will be to be aware of the context for vaccine procurement decisions during the pandemic this time, to be aware of the basic characteristics of the vaccines and some of the emerging issues. So this truly was a year like no other in so many ways. It was a time of great sorrow, and this picture on the left are Spanish healthcare workers who are gathering to mourn the death of a colleague who was caring for patients with COVID and died of it. At the same time, there were so many advances. When we look back, the acceleration of vaccine development has never occurred at this pace throughout human history. So the index case was on December 16th. Within 2 weeks, the genome of the infecting agent was known. The genome was uploaded to GenBank and virologic.org, making it available to the world's researchers, became declared a pandemic, and the first phase 1 trial started on the 16th of March. Over the summer, there were multiple phase 3 trials, and we know the results of those now with many stellar vaccine choices. And then a COVID-19 vaccine program begins in Canada. And so this is the time of great joy, sorrow and joy. This was the first participant in the UK program, a 90-year-old who received the Pfizer vaccine. So the vaccines that we have are in great part due to the procurement strategy for the Government of Canada. And as the current processes to bring vaccines into Canada are that they are generally authorized, and then provinces, territories, and the federal government decide to purchase them or jointly purchase them and use them in their jurisdictions. For the last pandemic in 2009, there was an influenza pandemic prototype vaccine strategy in place. And so that vaccine became available within months. At this time, there were no vaccines that were ready off the shelf to purchase. So the government put in place this vaccine task force, which I was very honored to co-chair with my colleague Mark Levenin, and a number of Canadian experts who were very motivated to help find a way to mitigate this pandemic. And our goal was to find a vaccine that would be safe and effective, whether it was domestic, international, and wherever possible, to link that with biomanufacturing in Canada to build our ability to produce vaccines and have those on domestic soil. Ultimately, this is the portfolio that was recommended to ministers. And you can see in the second column here, there's a lot of doses. There's many times more doses than we would ever need to give, even with a 2-dose strategy, 2 doses to every Canadian. And that's because at the time that these procurement decisions were being made, it was June and July. There were no phase 3 trial results. It was not known whether the companies that were starting trials would have the ability to scale up efficiently, whether it would work in terms of efficacy. And so the idea was to have a portfolio, and within each portfolio, 1 or 2 choices, so that in case one platform crashed and burned, there would be other platforms that might come through. Now, all of the strategies are based on developing an immune response to the virus, and a particular part of the virus, the spike protein shown here on the right. This is a conformational structure done by Jason McClellan, and who did the same for the SARS 2003 virus. And why is that an important immunological target? It's because the spike protein binds to the human receptor, ACE2 receptor. It has an S1 and an S2 subunit, which are involved in entry with an creation of an endosome using the host cell's envelope. Then viruses, as viruses always do, they either replicate in the cytoplasm or they go to the genome of the host. So we have RNA and DNA viruses, and then they just use the host cell as a factory to make viruses, which are then budded off to infect other cells. Now that turned out to be a very good strategy, this focusing on the spike protein. And this, I should say, was built on learning from SARS 2003. This graph, which is from a COVAX workshop recently, gives us two pieces of information. We're looking here at the leading candidates, which have phase 3 efficacy results. Generally, that's a month after the last dose of the vaccine. You can see on the bottom here the efficacy that was found in those trials. And then on the y-axis is wild type virus neutralization. So these are neutralizing antibodies, and the comparison here is to convalescent sera from humans that have had COVID-19 themselves. And the sera that you choose for these tests is people with a range of disease, mild, moderate, and severe. So you can see we're going from highest efficacy on the left to lower efficacy on the right. The Novofax subunit vaccine, 95% efficacy, and the neutralizing titer fourfold higher than convalescent sera. The Pfizer BioNTech and the Mrna, both very high, 94, 95%, and in the range of almost four-fold higher. And then lesser for these other strategies with the two adenovirus vectored vaccines, both of which are in Canada's portfolio, being 62% for Oxford and 66 to 72 for Jansen. Oxford has a comparable immune neutralizing antibody immune response to convalescent sera, and Jansen has 2 to 3-fold lower in those studies. So this slide shows us how efficacious the vaccines are by relying on the spike protein. It also shows us a correlation between neutralizing antibody and efficacy. This does not mean that it's the mechanism for the vaccine to protect us, and it doesn't mean that it's a true correlate, and that in fact has not been determined yet by the regulators, even though we do see this pretty consistently across studies. That remains to be declared. And perhaps Dr. Bettle will tell us more about that during her presentation. So we have 3 vaccines that are being rolled out in jurisdictions in Canada now. This is just copied from the National Advisory Committee on Immunization website, and you can follow the link there yourself. And you can see we have 2 mRNA vaccines, 1 non-replicating viral vector, which is the ChAdOx. The Jansen vaccine is I don't know why the title's not repeating there, is not being used yet, but is authorized as of March 5th. Its authorization is similar to the others in that it's 18 years or older. You give 0.5 ml, it's IM. What's different is there's only 1 dose. Like the AstraZeneca, it can be used at refrigerator temperature, doesn't have to be diluted, and has a similar time that it can be left at room temperature, but only 6 hours at refrigerator temperature, and it's multi-dose with a 5-dose vial. The mRNA vaccines are a little bit different volume, 0.3 and 5, and a little bit different age, 16 and 18, and our 2 doses. All vaccines are intramuscular, and the freezing, the temperature storage requirements for the mRNA vaccines are of course much lower than the other vaccines. I'll just put a little note here. Of course, we know that the advice regarding the age that can get the AstraZeneca product is different now, being the 55 is the lower age cutoff, and older, and some provinces are just giving up to age 64, as that was a previous recommendation. Now, of the portfolio, you saw protein-based vaccines, the Novavax, the Sanofi GSK product, and the Medicago virus-like particle vaccine. Those are the older platforms that we are all very familiar with as immunizers. I show here the routine immunization schedule for children and adults in my province, Nova Scotia. And you can see that for decades, we've been immunizing people with inactivated vaccines, live attenuated vaccines, toxoid-based, and subunit vaccines. So why aren't those first on the list for our portfolio? The inactivated vaccines, there was a concern that there would be enhanced disease if you provided the entire coronavirus molecule and just inactivated it chemically or by temperature. And so that was not a choice, but that is being used in other countries. That there was no live attenuated vaccine or toxoid, and then the subunit, we do have the 3. They, however, take much longer to scale up and to test. And all of the companies doing the subunit vaccines have had various obstacles to manufacturing at scale. So how do the mRNA vaccines work, and how do the adenovirus vectored vaccines work? The mRNA vaccines, just like RNA viruses, do their protein synthesis by using our own ribosomes to create the protein, which then sits on the outside of the cell, embedded in the membrane, to alert the immune system to make an immune response to it. So these are on somatic cells at the site of injection. And this is a lovely graphic from the New York Times. So you can see that the ribosomal transcription of the mRNA, it makes the monomer. The spike protein is actually a trimer. So that self-assembles and makes its way out to the surface of the cell. Adenovirus vectored vaccines use a DNA strategy. So this is the adenovirus representation here. There is a transgene in the adenovirus genome that encodes for the spike protein, so that when our cells incorporate the virus, just as they do for any other virus infection where the virus enters the host cell, it replicates at the site of the nucleus, making mRNA, and then it's going out, and similar to the mRNA vaccines, it's using the ribosome to make the product, which goes out to the surface of the cell. So that's how the Jansen and the AstraZeneca vaccines are made. Now, I showed you the slide that showed the various efficacy, with the highest efficacy being in the mRNA vaccines. One of the common public health messages you see across all provinces and countries really is the efficacy against death and hospitalization, which is 100%. So even though there may be varying efficacy against laboratory confirmed disease, which according to the trial may be what we might consider mild or moderate, the efficacy against death and hospitalization is stellar. And that's usually the goal of a public health program, to prevent the most severe outcomes. Now, I know Dr. Kwong will be speaking about surveillance, so I only have one slide on the adverse events characteristics with these vaccines. Just a reference on the bottom right of your side, there is standard terminology for describing something as common and rare and so on. And again, a reminder to those not in the vaccine world, adverse events are any untoward medical occurrence that follows immunization. It does not mean they're causally related. So overall, the most common adverse events are pain at the injection site, headache and fatigue, and very rare, the two most common that are not common, they're actually rare, are anaphylaxis after the Pfizer vaccine with about 11 doses per million, and then the clotting and low platelet disorder after the AstraZeneca vaccine, which depending on the jurisdiction is anywhere from 1 to 125,000 to 1 in a million doses. And lastly, what are the issues on the horizon? We're not done yet. We have the emerging variants. with different disease severity and may affect vaccine performance. That story is still being written. We have to plan for the various different trajectories the virus might take over the coming years. And of course, we have the rollout to do with varying capacity across jurisdictions, vaccine hesitancy, and still potential interruptions to supply. The most recent newsworthy item being the problems that occurred in the emergent plant with regard to the Jansen vaccine. And while we do this, we have to maintain public health measures, of course. So I'll stop there and pass on to Dr. Bettle.
That's great. Thank you very much, Joanne, for that. And you just given up the screen there, and then Dr. Bettle will take over the screen, I think, here.
Okay. Oh, stop. New share? No.
Oh, you have to, yeah, that's right. Okay.
Right. Okay.
Over. Okay, does that show properly?
Yeah, we can see it just fine. That's your second slide. That's perfect. Thank you very much.
All right, so thanks very much. So this is, I believe this is my second CanCOVID presentation. I think I was here last summer talking about Health Canada processes to expedite the review of clinical trials for COVID. So it's really exciting to be able to come back to the same group and kind of talk about what we've done since then and what we're doing to actually authorize COVID vaccines and other products as well. So for this audience, I mean, this is a pretty basic slide, right? But it just kind of wanted to give a reminder of normal processes for developing vaccines. As Joanne was indicating, this has happened really, really quickly. So in a normal world, over a couple of years, you would have a number of candidates identified, all of the non-clinical work would be done, and then you would have several phases of studies, generally with breaks in between, and kind of a slow build up to assess the safety and efficacy of the vaccine. And at the end of all of this, when all of the data was collected, the manufacturer would then come to Health Canada and make an application for review. However, obviously, this hasn't happened in the pandemic situation. Manufacturers have scaled up very quickly. They've developed their manufacturing at risk as clinical trials were going on. And they've used adaptive clinical trial designs that have allowed them to go really quickly from initial phases to scale up to larger and larger populations. Still want to emphasize to people that the clinical trials have been done, and they've been done in tens and thousands of people, and the regular animal work has been done, but people have tried very hard to compress these steps. Similarly, Health Canada has looked at our processes through all of this, and our normal process of waiting for all of the data to be acquired, get a submission, and spend 10 months reviewing it isn't suitable for the current context. So I'll talk a little bit about what we've done to speed this up. And what we've done really is introduced a number of interim orders. So an interim order is a temporary regulatory tool that's able to be put in place really quickly, so much more quickly than you normally would be able to modify regulations. And there was one in particular I wanted to talk about, which was put into place last September. The interim order lasts for a year, and this particular one introduced a number of flexibilities. The first is rolling submissions, and I'll talk about that on the next slide. Some more flexible data requirements, so reducing some of the more administrative requirements on manufacturers. And the introduction of the ability of Health Canada to put binding terms and conditions on an authorization to require additional evidence submission from manufacturers. We also started working very early with manufacturers before they had started their phase 3 trials, before they were thinking about coming to Canada. So we proactively reached out to a lot of the major manufacturers who are also talking to the Vaccines Task Force that Joanne was referring to start talking to manufacturers really early and to provide advice about what we would want to see in a Canadian drug submission. We also worked very closely with our international regulatory partners. So all of the major regulators have been meeting basically weekly to talk about scientific issues, to develop a fairly aligned approach to vaccine submissions, so that when vaccines were ready, they could be reviewed and authorized very quickly globally. So what does a rolling submission look like? As I said, normally we would have all of the data come to us at once. The idea of a rolling submission is that as manufacturers develop their evidence, as they begin to scale up their manufacturing, they could start to flow that information to Health Canada. And so we could actually review it in real time. We had manufacturers come to us with a basic outline of when they would provide us what data. And then as they were going through, we would work with them really intensely so we could plan our resources to be able to review the next piece and to be able to anticipate when we'd have the next piece. We anticipated it would take several months to review the complete data from the manufacturer, but all along there was this plan that throughout the review, we'd be defining what was enough to make an authorization decision and what our knowledge gaps were. And that's really the terms and conditions piece. So identifying studies that needed to be completed, evidence that needed to continue to flow in after authorization, and just to make sure that we were very clear on what was needed to make sure that we were confident of the safety and efficacy and quality of the product. So an authorization decision is really based on the integration of all of the evidence that you have. Are you confident that the product is safe and effective? Does it work the way you expect it to? What do we know about the risks, and what do we know about how we can mitigate them? And where we have further questions where there might be potential risks, what's the plan to continue to gather that data and to characterize those risks so that we know, are there additional populations at risk? Are there people who absolutely should not get the vaccine? And then part of the review process is also making sure that we can provide all of the information to Canadians and to healthcare professionals to accurately reflect what we know about how the product should be used. So using terms and conditions is a new authority for Health Canada. We have had for a long time a notice of compliance with conditions policy, which is kind of a conditional authorization, where when there's promising evidence, we could let a product come to market with the requirement the manufacturer continue to provide data from ongoing studies. And that's most commonly used for drugs for rare diseases or cancer therapies, where you might have promising evidence and you would want to provide that product to patients, but also continue to flesh out the evidence. With terms and conditions, it's a much stronger legal hook because it's tied to the authorization, so that if the manufacturer doesn't meet those conditions, we could suspend the authorization or remove it. So it's kind of a strong binding requirement. So during the course of the review, as I said, we would identify what studies were ongoing, where there were gaps in our knowledge, and develop those terms and conditions which were required before a product authorization could be granted. So throughout the course of the review of all of the COVID products, we're also planning for post-market activities. Part of the pre-authorization review is the review of a risk management plan. And a risk management plan is a defined report. It's common across a couple of different regulators, where basically the manufacturer is required to identify everything we know about the product. What do we know about the safety profile? Where are there gaps or questions, and are there potential issues that are in need of further characterization? And then the manufacturer is required to propose a pharmacovigilance plan and other studies so that all of these gaps in our knowledge can continue to be filled in. Once on the market, manufacturers are required to report on adverse events to Health Canada. And in the vaccine space, there are actually multiple streams of data. So on the public health side, healthcare professionals can report adverse events to their local public health unit and to their provincial or territorial public health officials. And then those reports and that data is brought together with the data that Health Canada has, and we work very closely with the Public Health Agency to assess the safety situation and determine whether any further actions are needed. We're also, again, in the post-market space, working very closely with our international partners so that should new issues emerge, we're talking regularly, several times a week, to discuss these cases and to determine if any actions are needed. So one thing that's been really important throughout the COVID process, and especially in vaccines where there is a lot of misinformation about the use of vaccines, we want to make sure that we're making information available and being very transparent so people can understand how decisions are being made to bring products to Canada. So we have developed a whole new resource for COVID products, which we call our COVID portal. And when a vaccine is approved, we're making all sorts of information available so that people can look at kind of summaries of the evidence that we've reviewed. They can look at the terms and conditions that we're imposing on the manufacturers. And then we're making additional information available as we go along as well, including all of the clinical trial information, which was reviewed as part of the drug submission. So a month or so after our initial authorization, that data is becoming available so that people can look, if they're so inclined, they could go in, they could analyze the clinical data themselves. And there are also more detailed descriptions of all of the information which is used to make the authorization decision. So when we first started this, the first question was, okay, you've got this interim order, you've got dedicated teams set up to review vaccines. How long is it going to take? And more importantly, how quickly can you do it to bring these products to Canadians? And I would say every vaccine review has been very different. So each manufacturer has come to us at a different level of readiness and with more or less precise plans on when they were going to file that data. Sometimes the clinical trials have taken longer than expected to read out or to recruit participants. Some of the trials have been paused for safety reasons and then restarted. And what we found most challenging is as manufacturers have been scaling up to meet the demands of the entire world, they have sometimes run into difficulty, and it may take longer to establish new manufacturing sites or supply chains. So that's been a variable in the time it's taken to complete all of our review. And then we've also found that manufacturers have been dealing with multiple regulators all at the same time. So that's also a challenge for their resources as well. However, what you can see here is this is kind of an overview of everything we've done under the clinical trials, or sorry, the drugs interim order. So since we got our first COVID vaccine submission on October 1st last year, and we approved our first one on December 9th of last year. So we've now, depending on how you count them, we've approved 5 separate vaccines. From a regulatory perspective, we count the Serum Institute of India version of the AstraZeneca vaccine as another one in our list. But whatever way you look at it, most of these reviews have been completed in 2 to 3 months, whereas we would normally have a 10-month review timeline for the completion of a new drug submission. We still have one additional vaccine under review right now, which is the Novofax Vaccine, and we can still anticipate further vaccines, which are a little bit later in development. We can still expect some additional COVID vaccines to be submitted to Canada. We also have a number of treatments, we have 2 which have been authorized and a number which are still under review. And a wide range of treatments as well, anything from antivirals to quite a few monoclonal antibodies, which will still be useful. I mean, we still have obviously quite a lot of disease in the country, and we still need treatments for people who develop serious COVID-19, as well as the vaccines. So what happens next? So obviously we have to keep reviewing the submissions that we have currently under review. We have found that for all of the initial vaccines that we've approved, we've had multiple follow-up submissions for new manufacturing sites, updates to the packaging and the labeling or the numbers of doses in the vials. And what we're kind of eagerly anticipating will be the submissions that should come soon to expand the indications. So we know that all of the vaccine manufacturers are running pediatric trials or adolescent trials, and some of those trials are starting to read out. So we can expect those submissions to come in very soon. And those reviews will also be expedited as quickly as we possibly can so that we can make these products available to kids hopefully through the summer so they can go back to school in the fall. everyone will be aware that as the vaccines are being rolled out to millions of people around the world, that's when we're starting to see these very rare potential safety signals. So our focus is also switching to the review and assessment of those and our continuing collaboration with our international partners. Looking ahead, we are preparing for when we might need a modified vaccine to address those variants of concern. And Health Canada has recently issued a guidance for manufacturers in collaboration with our partners in the UK and Australia and a couple of other countries about the data requirements that would be required for that. So we would anticipate submissions for a modified vaccine to be fairly similar to the process which is being used now for flu vaccines. So a strain change rather than having to go back to start from scratch with very large clinical trials. Finally, as I mentioned at the start, these interim orders were just transient tools. So we need to, it's more of a legal mechanism, but we need to transition all of these products which have been authorized under the interim order into full authorizations under the Food and Drug Regulations. So this provides an opportunity for manufacturers to come back to provide the data that they have accumulated to now as part of that submission, and then we can make that decision as to whether or not they can be continued with a full authorization. And I think that's it, so I'll stop there, thanks.
Okay, thanks very much, Megan, for that concise and comprehensive overview. I'll just state that I'm glad to see you, as speaking as a pediatrician, I'm glad to see you mention the part about vaccinating children this summer. Next, we'll turn things over to Dr. Carolyn Quach, who will join us for her presentation. She's taking the screen, so I'll turn things over to you. Thank you.
So thank you very much, Jim, and thank you for the invitation. So I was asked to speak about formulating public health guidance, the NACI process, recommendations, and rationale. I have no declaration of interest except from the fact that I am the chair of the National Advisory Committee on Immunization for another 64 days, not that I'm counting. So I think Dr. Bettle just summarized what Health Canada did, but I wanted to also maybe contrast the difference between the roles of Health Canada and NACI. So NACI is a NITAG, which is a National Immunization Technical Advisory Group, which is similar to SAGE, for instance, that is giving advice to WHO, or ACIP that is giving advice to the CDC. So NACI gives advice to the Public Health Agency of Canada. We are both a technical resource and a deliberative body that empowers the national authorities and policymakers to make evidence-based decisions. We are a resource that is particularly important in view of the complex and vast bodies of evidence and the global interdependence and integration of health systems. That's what Dr. Duclos, who finished his career at WHO, said in 2010. So if you look at the difference between the regulator review and our NACI advice in terms of purpose, so Health Canada will authorize specific indications for use that are expected to be safe, immunogenic, efficacious, and of suitable quality for individuals. We will recommend vaccination strategies to promote health, prevent and control infectious diseases, and prepare for or respond to public health emergencies. In terms of focus, Health Canada will look at individual use of product, whereas NACI will look at use of product for public programs and population health, and sometimes for individual recommendations. But during COVID, it is really only looking at public health, population health, and public programs. I think the other difference is that Health Canada will review each product independently, whereas NACI has to rank products one versus the others so that we give some, I'd say, guidance to provinces and territories as to how to use the various vaccines, mainly when, like in COVID times, we have more than one. The data that Health Canada reviews are preclinical and clinical trials data with manufacturing information submitted by the manufacturers and post-marketing monitoring. NACI will review all relevant and available evidence for specific vaccines and similar vaccine formulations in the context of public health considerations. So we do not look at the preclinical, we will look at clinical trials. We will also look at real world evidence, so effectiveness once they become available. We will look at vaccine programs and schedules, disease burden and distribution, and outbreak management data. The thing that seems to be a little bit confusing is that we both, Health Canada and NACI, are under the Minister of Health and the federal government, but there is no hierarchy of one versus the other. We work side by side and we talk to one another, but one doesn't tell the other what to do. I think one other important point is that NACI can make off-label vaccine recommendations. We have made it in the past. I think it hasn't just been noticed because we weren't so much under the spotlight, but we've made multiple off-label recommendations. And we do that when there's a clear need supported by a public health ethics analysis. So NACI was established in 1964 by the Government of Canada, at the time Health Canada. It provides public health advice relating to immunizing agents used for the prevention of disease in humans. The scope was traditionally only looking at recommendation based on safety, efficacy, immunogenicity, effectiveness, and burden of illness. But since 2016, our mandate had been gradually expanded to also include programmatic factors such as program feasibility and cost effectiveness where relevant. We operate as an external advisory body to PHAC, which means that we are all volunteer people who are selected based on our expertise, and we are arm's length from PHAC. So we make recommendations to PHAC. PHAC could decide not to implement them or accept them. And then these recommendations are sent to provinces and territories, but are non-binding. They are really there as guidance. So these are the elements that we looked at. And I'm going to use the extended interval example to go through them because I think that that has raised a few questions. So the first thing that we look at is the burden of disease. In the particular case of COVID and other diseases, these burden of illness are provided through the provinces and territories surveillance programs that are then brought back up to the surveillance branch of the Public Health Agency of Canada. So the data we look at are outside of NACI. We also will do literature reviews to have evidence of burden of illness. In the particular case of COVID, we have decided not to update continuously our reports, but rather refer to the sites from the Public Health Agency of Canada where that burden of illness is captured. We then look at efficacy of vaccines, which means data from clinical trials. And that's usually where we start, at least with COVID, because that's what was available to us at first. And so if I take the example of the AstraZeneca, when we made our recommendation to stop at 64 years of age, while Health Canada decided to approve it for all age groups, at the time where we made our recommendation, the only data we had were the clinical trials data, which showed that in the 65 plus, the confidence interval around the point estimate, which was around 40%, was very wide and including 0. In that particular case, we also had one real world evidence, which was still a preprint from a Scottish study for which we had issues with methodological biases. And so we decided not to change our recommendation based on that one real world evidence. Of course, then in the next 2 to 3 days after the recommendation was published, there were 2 additional real world effectiveness data, pre-publication papers that were made available. And at that point in time, we reassessed the body of the literature to then change our recommendation to allow the utilization of the AstraZeneca for all age groups. So that's efficacy and effectiveness. So we monitor continuously the what is being printed. And of course, with COVID, as Jim says, there is more papers that are printed in a day than one can master. So we have a team that does that day and night, but we of course will not act upon one new preprint. We wait for a few to accumulate to then review the body of evidence. Otherwise, it would be absolutely inefficient. We, of course, look at immunogenicity of vaccines and data. So all the studies that came out on the fact that elderly and immunocompromised individuals had a lower antibody response, we of course then balance that out with effectiveness data when we have them. And that will be also looked at from even preprint. So even before we have access to the data, we will talk to various groups, including the UK, BC, Quebec, where surveillance is ongoing and where extended intervals have been used. And so that's how we sometimes change our recommendations. We will also look at safety, so data that are coming back from the various regulatory agencies, other NITAGs around the world, published data, again, our surveillance systems, and I think Jeff will talk about that later on, so I will not go into other surveillance systems, but we have access to all those data, and again, we have presentations from various groups when there are new data emerging, at least in Canada. We have a link with the public health ethics group that reviews our recommendations and makes sure that all ethical elements are taken into consideration. They sometimes make us, recommendation for change in either wording or how we see things. So that is actually quite helpful. We ensure that the program is actually equitable so that the decision that we take, let's say to favor one group, is not going to be at a systemic disadvantage to other groups. And we have an EFA framework. EFA stands for Ethics, Equity, Feasibility, and Acceptability that we follow to make sure that we are not providing recommendations that would not fit with the NACI's mandate and goals. We have cost effectiveness analysis for some vaccines. In the case of COVID, we decided that cost was not an issue, so that whatever the cost, we would still move ahead with the program. So that hasn't been looked at. Feasibility, of course, given the resources available, which for COVID, again, we would just have to make it work. And acceptability, where we don't do our own acceptability surveys, but work with other programs, and we are the knowledge users for various research grants or surveys that are done by the Public Health Agency of Canada. So if we take the extended interval, the elements that we actually looked at were the effectiveness of the programs where extended interval were used, mainly in the UK, where a 12-week interval was utilized from the get-go. In Quebec, where at least 12 weeks was used from the get-go in all population, including long-term care facilities and healthcare workers. And in BC, where both Quebec and BC said that by the end of March, knowing that the program had started towards mid-December to end of December, the vaccine effectiveness was still at close to 80% for symptomatic diseases, hospitalizations, and death. So that's the effectiveness data that we've looked at. We have also, as I said, looked at immunogenicity data, but had put more emphasis on effectiveness data. And so if you want to have access to all the data we reviewed, the last NACI statement that was published yesterday runs through all the elements. And then we use a mathematical model that I'm showing here, where we inputted the effectiveness point estimates from the various studies in the various age groups, where we used a decrease in protection between 1%, 4%, and 8%, depending on with the sensitivity analysis, with an interval going from 6 to 24 weeks. And what that shows here is that if you were to look at hospitalization and death in the Canadian population, modeling a third wave of variants starting early April, the scenario that would give you the best gains in terms of decrease in terms of hospitalizations and death was using a 24-week interval. However, we had data up till 10 weeks when we looked at this, and we had data from expected supplies in Canada, which showed us that the maximum interval that would be needed was 4 months. And so the recommendations that NACI decided to make was to vaccinate as many people as quickly as possible to have a protection that was shared among people, because this 80% protection or even 70% protection was a scenario that would decrease which would have the best gains in terms of public health goals, keeping in mind that the objectives of this pandemic preparedness are to decrease or to protect the vulnerable population directly and indirectly, and to decrease societal disruption. And the goal from the vaccination program is to vaccinate as many Canadians as possible. These are not goals that NACI made up, but these are goals that were agreed upon by the Public Health Agency of Canada with provinces and territories. So we don't have all the data, but we have at one point in time to make a decision. The extended interval decision was made because provinces and territories asked NACI how to best use the vaccine supplies that they had at hand for the maximum goal and gains in terms of population health. So NACI's role in this FPT process, so we were identified in the FPT pandemic strategy as the authoritative body for advice on vaccine prioritization and vaccine public health program design. So we would provide expert advice and guidance for the use of pandemic vaccine, including prioritization. Our recommendations would include dosages and schedule based on pre-pandemic studies of relevant prototype vaccine, and to the extent possible, rapid clinical trials of new vaccines. We used to meet 3 times per year. Now we're meeting every week. We have expert working groups that will develop draft products for NACI consideration and voting. At this point in time, everything has been folded together because we meet every week, all of us. The working groups are chaired by NACI members. NACI will submit its advice to the Public Health Agency of Canada for consideration and approval by the Vice President of the Infectious Disease Prevention and Control Branch. PHAC provides the scientific project management and logistical support, and the NACI Executive Committee is composed of the chair, the vice chair, the executive secretary, the secretariat manager, who provides oversight on meeting agendas, methods, work plan items, and priorities. We are a committee of 15 voting members, plus the chair who doesn't vote. And we have liaison members to the 3 organizations who are involved with vaccination. I think that one thing that needs to be made clear is the roles of provinces and territories. So you know that in Canada, health is a provincial jurisdiction, that provinces and territories administer and deliver healthcare services, including immunization-related programs. They have the responsibilities of immunization policies and schedules. They have each expert immunization advisory committees, or at least they should. They are responsible for purchasing vaccines, except for COVID, which is purchased by the federal government, design and maintain immunization registries, do surveillance and program monitoring, and do public and professional education and engagement. But each jurisdiction has its own process and mechanism for setting immunization targets and planning, designing, implementing, and evaluating immunization programs. For COVID, the decision to extend the interval or not, or to stay to, with whatever was decided before, is of provincial jurisdiction. NACI has no binding recommendation. We have no power whatsoever. We are just there to make suggestions and recommendations that then have to be uptaken by PNTs. And so PNT governments typically initiate their vaccine and immunization program assessment processes following or in tandem with NACI, and where applicable with the Canadian Immunization Committee. So thank you very much. I'll stop sharing my screen.
Okay, that's great. Thank you very much, Caroline, for a beautiful overview and helping distinguish the 2 sides of it at this federal level. So our final presentation before we get to hear from Dr. Talbot is Dr. Jeff Kwong is going to talk about vaccine surveillance in Canada. You've got the screen there. Jeff, thanks very much.
Great. Okay, super. Yeah, so I've been asked to talk about vaccine surveillance in Canada, and here we go. And now it's not advancing. Sorry, I'm having some.
Sometimes you have to start by going the bottom left corner there, and then it.
Okay. Let's see if this works. Okay, there we go. Okay, super. So I have no financial conflicts of interest to disclose. So I just listed out here a few components that would be usually what we would consider for vaccine surveillance activities. And I put in parentheses, they're no less important, but I'm just not going to talk about them today. And those are vaccine confidence and then program impact and evaluation. And I'm mainly going to focus on vaccine coverage, safety, and effectiveness. Okay. So just as an example of coverage, this is from the PHACS website. And you can see a coverage as of March 27th, which was almost a couple of weeks ago, but that's the latest data available. The cumulative percentage of the population who have received at least one dose of a COVID-19 vaccine. And you can see that there are some variations by province. Some are higher than others. And hopefully, as of today, they're actually a lot higher than the numbers here. In terms of vaccine safety, there's this was alluded to by both Joanne and Caroline, that there's passive surveillance. So there's these reports of adverse events following immunization, and they can be reported by healthcare providers as well as vaccine recipients. And they would go to local public health departments, which would then investigate them and then report to provincial public health authorities and then and onwards to federal bodies. So these are, the bread and butter of public health work in terms of vaccine safety surveillance. And so they would be investigating reports of, anaphylaxis that occurs after vaccine receipt, and other APHEs. And then second component is that there's active surveillance. So there are a number of networks that do active surveillance. There are a couple of hospital-based networks. One is called IMPACT, or that's Immunization Monitoring Program Active. And that's a network of pediatric tertiary care hospitals that do vaccine safety surveillance. And then more recently, there's this Serious Outcome Surveillance Network, which is part of the Canadian Immunization Research Network, or CERN. And they are a network of adult hospitals that also do hospital-based surveillance for vaccines. And then another network is known as the CANVAS, or the Canadian National Vaccine Safety Network. And so this is a participant-focused network where they use an app to collect data on vaccine recipients and people who have not been vaccinated as well as a control group, and they will ask them, within this, like they'll do a survey 8 days after vaccine receipt. Have you had any side effects from the vaccine? Have you had to take time off from work or school because of a side effect? And so everyone can register for this. You can go to this website, canvas-covid.ca, and whether you have received or not received the vaccine, you can register. And so, this is similar to, in the US, there's a system called vSafe, but the difference is that the Canvas platform is also enrolling control subjects so that we can have a baseline to compare against. And people who don't yet get vaccinated and are controls can subsequently sign up after they get vaccinated and then be one of the vaccinated cohort. And then lastly, there are special studies. And so that's what I'm calling hypothesis testing studies that are based on safety surveillance system signals or RCTs, where we see that maybe there's a side effect from vaccine, and let's confirm if there really is, if it really is associated with, if this particular adverse event is really associated with the vaccine or not. And I'll be talking more about how we do those studies. And there's also the Special Immunization Clinics Network, which is also another CERN network, which does investigations of people who have had serious APHEs, after immunization. Okay. Now for vaccine effectiveness, here's just some data from Quebec, INSPQ. This was put out in February, there was some preliminary estimates of vaccine effectiveness in healthcare workers and then in residents of their long-term care facilities. And you can see that, after in healthcare workers, 14 days, it was 74%, and then it went up to 80% by 21 days and beyond. And then this is after the first dose. And then for long-term care residents, they got 80% vaccine effectiveness after 21 days following vaccination. And so there's, it's really important to do these real world studies of vaccine effectiveness, not just for long, lab confirmed COVID infection, but also for more serious outcomes. And I'll be talking more about that. Okay. So, so the network that I co-lead for CERN is called the PCN, or Provincial Collaborative Network. So CERN was created in 2015 to study to do like kind of a bunch of vaccine related research in Canada. And the Provincial Collaborative Network, we do studies basically using provincial databases. And so we obtained funding from CIHR, like CERN obtained funding from CIHR. And we're also obtaining, in the process of obtaining funding from the COVID Immunity Task Force to conduct post-market studies of COVID-19 vaccines. And so this is a collaboration between 5 provinces at the moment. So we've listed the 5 provinces here, the institutions from each of the provinces. And you can see that the population, we cover a total of 34 million Canadians, representing about 90% of the population in Canada. And so I'm going to talk mainly about the infrastructure that we have in Ontario. I'm based at a research institute called ICES. And this is just a schematic of the data infrastructure that we have. I'm just going to spend a minute just to go over this, just to give you an example. And then this is kind of replicated in the 4 other provinces. So in Ontario, we have a database called the Registered Persons Database, which is basically a registry of every Ontarian who's ever had a health card number. And we use something called the ICS key number, or IKN, as a linking, as a unique identifier for each person. And we can link to all the records, so for instance, the health administrative data, physician billing claims. So every time you go see a doctor, the physician submits a claim to the government for seeing the patient. So that claim, will have the health card number, and then we can link to the IKN, which is basically an encrypted version of your health card number. We can link to like all the drug claims, emergency department visits, hospitalizations, et cetera. And then here on the right-hand side, we have other data sets like the cancer registry. We can identify immigrants with linkage to this Immigration, Refugees and Citizenship Canada landed immigrant database. And we have other derived cohorts where we use like algorithms applied to the administrative data to identify people with chronic conditions such as diabetes, asthma, COPD, et cetera. And then we can also link to other data sets. So this is definitely not a comprehensive list of all the data sets that we have, but this is just an example of some of the data sets that we can link with using this IKN that we have in Ontario. And then we link the databases that are necessary to create research data sets, which we use to conduct research studies. Okay, so for the COVID studies that we're going to be doing, the most important element obviously is going to be the COVID vaccination data. And so in each of the provinces that are participating, we are receiving the COVID vaccination data either on a daily basis or a weekly basis or a monthly basis, but we do have access to basically records of all the COVID vaccines administered in each province. We also have access to the laboratory testing data for COVID. So, whether, mostly it's PCR. There may be some serology in there and some rapid tests, but mostly it's the PCR testing. And then we also have the outcomes or health services use, and we can look at mortality, hospitalizations, including ICU admissions, emergency department visits, and also physician office visits. And then we can look at various populations of interest. So we can look at residents of long-term care, age groups, so whether the elderly or children, those with chronic conditions. On the next slide, I'm going to go through some of the chronic conditions that we are able to identify. We can look at pregnant women, immigrants and refugees, Indigenous populations, at least in Ontario and Alberta, and then for the homeless population using algorithms applied to the administrative data. On the right-hand side, I've listed geographic regions. So we'd want to know what, for instance, coverage is in smaller levels of geography, not just like the province, but also at, the public health units in Ontario or, other smaller units of geography. And then we can use census data we can use that as a proxy for characteristics of individuals. So, so for instance, we can use the dissemination area, or DA, which is a very small level of geography. It's only 400 to 700 people on average. And we can use that as a proxy for individual level measures, to identify, areas where there's more essential workers, so people who have to work outside the home, larger household sizes, people with lower income or education, visible minorities, and recent immigrants. And then for the chronic medical conditions, we can identify this list here. So you can see there's various chronic respiratory diseases, cardiovascular diseases, diabetes, cancer. And here on the right-hand side, you can see we can identify people who are immunocompromised, so whether they have HIV, they've received a transplant in the past, or they're on immunosuppressive medications. People with autoimmune diseases such as rheumatoid arthritis, inflammatory bowel disease, multiple sclerosis, and lupus. And then people with chronic kidney disease, liver disease, dementia, and people who've had a stroke or transient ischemic attack in the past. So these are just some examples of some of the conditions that we will identify, and we can look at, we can do vaccine effectiveness studies or safety studies in these various populations. Okay, so here's just some examples of some of the stuff that we've been working on so far, and then I'm going to talk about some stuff that we've got planned. So in terms of vaccine coverage, this is an example that we just put out last week for the first time, and this is updated data that's coming out, I think, tomorrow morning. So what we've done here is we've taken the population of Ontario and we grouped them into 10 deciles. So decile 1 to decile 10. And we did this by looking at the FSA, or the forward sortation area, which is the first 3 digits of your postal code. And within each FSA, we looked at the cumulative incidence of COVID-19 infections that were lab confirmed. So number 1 here is the areas where there've been the highest incidence of COVID infections since the start of the pandemic. And number 10 is the lowest incidence of COVID infections since the start of the pandemic. And then what we did is we stratified it further by age group. So we have the 80 plus and a 5-year age bands all the way down to here, where we had a bigger group here of 15 to 39. And we looked at the vaccine coverage. And this is data as of April 5th in Ontario. And you can see here that coverage for the 80 plus overall was 73%. But so ideally what you'd see is that the areas where there's highest risk is where you would hope that you would have highest vaccine coverage, because they're in greatest need. Because we know that historically they've had high rates of COVID infection, so you'd hope that we'd have higher rates of coverage. Unfortunately, what we see here is that we have the highest coverage here in decile 10 and the lowest in decile 1, at least for the 80 plus, and with the the difference of 18 percentage points from lowest to highest. And then and so on, down. You can see here that maybe in some of these older, like 60 to 64 and 65 to 69, we are doing maybe slightly better here than in the deciles 9 and 10. But you can see in the younger age groups under 60, you can see that the red is bad and yellow is better, and you can see it's more red here in the lower deciles. And you can see overall lower coverage in decile 1 compared to decile 10. So this has led to more attention to try to reach out to these high-risk areas. And so we've had a shift in the rollout in Ontario, where we're going to try to vaccinate, turn more attention to these high-risk areas. So I think this has been helpful, and we can maybe do this in other provinces as well. So an aspect of safety is to look at background rates. And so the Brighton collaboration had a list of adverse events of special interest that they generated well before vaccines were released. And these were outcomes or conditions that may or may not be related to vaccination. Some of them are actually, we think that are related to COVID infection. So, these are things to watch out for in case they may also be associated with vaccination. And so they came up with a list of these ACEs And so we've done some work, this is work that we put out as a preprint, I think last week. And we were looking at these thromboembolic and coagulation disorders, given all the attention with the AstraZeneca vaccine. Unfortunately, this is not the same as the VIPIT, but at least this gives you a sense of, these selected thromboembolic and coagulation disorders. So if you're interested, you can go take a look at this paper. We looked at outcomes like stroke, deep vein thrombosis, pulmonary embolism, as well as cerebral venous thrombosis. And we looked at it in hospitalizations and emergency department visits, in 5 years prior, 5 years prior to the pandemic, as well as 2020, just to get a sense of what the rates are of these conditions. And then we also looked at, this is another preprint that was published posted a few weeks ago, where we looked at background rates of all-cause mortality, hospitalizations, and emergency department visits in the long-term care residents in Ontario. Given that the rollout happening there, we were just wanted to see what the rates of these severe outcomes were. And so you can see here, this is like 10 years of data prior to the pandemic. You can see this is the number of long-term care residents in each month. And you can see it's pretty stable. And then in 2020, this big red line here, you can see that it went from 84,000 at the start of the year down to about 74,000 by the fall. So you can see there's a huge drop in the number of residents. And then here you can see that there was a large spike in mortality in April and May. And then it went back down to about the baseline after that in June and onwards. And so you can see that there's, high rates of mortality, each month about 3% of long-term care residents would be expected to die. And you can see there is some seasonality. So in January, and it would be higher than in the summer. So, this was something that we wanted to watch out for, when we were rolling out the vaccination program in January in the long-term care sector. And then looking at hospitalization rates, you can see that there's a huge drop in hospitalization rates with wave 1 during March, April, and it never went back up to the baseline, at least not until, at least up till September. And then emergency department visits, also a huge drop during this wave 1. And then it seems to be heading back up, but don't have the data to see if it has gone back up now to the baseline or not. And this is one of the problems with administrative data, is that there's lags in availability. And so we can't do a lot of stuff necessarily in real time with administrative data. Okay, so some of the planned safety studies, so that, the clinical trials, they would vaccinate, tens of thousands, but they also excluded certain populations. So we can look at, once we get into the real world, for instance, we can look at safety in pregnant women and safety in people with autoimmune disorders. How safe are they? Are they having, more events compared to, non-vaccinated people? And so we'll be monitoring this in the coming months. And we can also look at severe and or rare adverse events using a design called the self-control case series design, and where we use each person's own control. We've identified people who have been vaccinated and had the event. And we're basically looking for a clustering of the adverse event following immunization compared to control periods. And so we've done this design has been used for a lot of vaccine safety studies, and we can use this for COVID vaccine. But we'll need time for these events to occur before we can do these studies. In terms of effectiveness studies, we'll be generating product-specific estimates. So for the different vaccines that are currently available, we'll definitely be looking at the question of full versus partial vaccination. So the people who've only had one vaccine so far, does the vaccine effectiveness decrease as we get out to the 12 and 16 weeks? And how does it compare to people who have received 2 doses already? We'll definitely also be looking at the variants of concerns to see if vaccine effectiveness is lower against these new variants compared to the original strains. We'll look at severe outcomes like hospitalization, ICU admission, and death. We have to wait for the administrative data for that to happen, or we can actually use some of the surveillance data, which is where hospitalizations are recorded, along with cases. We can look at the impact of previous infection to see if people who had previous COVID infection, because we have records, at least if you've had lab confirmed COVID infection, we could see if VE is different in that population. And then, we can look at populations that were excluded from RCT, such as immunocompromised, to see if what VE is in that population. And then lastly, in long term, we can look at duration of protection. Does it last for 6 months, a year, or longer? How long do the COVID vaccines protect you for? And so that's it. This is our study team so far, and and growing, and so happy to have other people. If you're interested in helping with some of these stuff, we'd love to. There's tons of work to do, as I've alluded to, and so we'd love to have help with this moving forward. Okay, happy to answer any questions.
Okay, thanks very much, Jeff. And you can stop sharing screen. That's great, thanks. And we're going to turn over now to, after all these great presentations, to Dr. Jim Talbot, who is going to pull it all together for us in a few minutes and call on his decades of experience as a public health microbiologist and public health provincial medical officer of health leader to offer some comments. And time marches on. The session, the workshop will be over at 1:30, so we won't likely have all that much time for questions. There's been a lot of very thoughtful questions put into the chat box, and if we get to some, we will. But turn things over to Dr. Talbot right now. Thanks very much. And do you want to take control of the screen there, Jim?
No, mine's just purely verbal.
Okay.
There's too much material to cover with slides. So, the first thing I want to do is thank each of the 4 presenters. I managed to get the AstraZeneca vaccine, my first dose 2 weeks ago, and one of the emotions that I had that I wasn't expecting was a profound gratitude for everybody who'd made that moment possible. And I think all 4 of you, the work you've described, the people who are involved, were part of that. So on a very personal level, thank you. I will say that I think the theme of the talks that I've heard so far is encapsulated in the phrase, the problem with doing the impossible often enough is that it becomes part of your job description. So why do I say that what's been described is impossible? Well, I'll take you back in history to the early 1990s, the first consensus conferences the federal government convened on pandemic influenza, our state of preparation and the planning that was going to be required. And I attended the first one as the director of the provincial laboratory for Alberta. And I remember distinctly walking out of that meeting thinking that in terms that only make sense if you grew up in northern Alberta, that we as a country were hooped. There was nothing in place for us to do well. The surveillance systems across the planet didn't work very well. They were leaky. There were big patches where there weren't any. There was a lot of technology that was required that was very unevenly distributed. Some of the challenges that we faced around things like both the labs and vaccination had to do with the potential that an influenza vaccine would have to be grown up in eggs, and if it had avian characteristics, it would kill the eggs, so we wouldn't get enough virus to be able to make the reagents we needed in the lab, and we wouldn't be able to make vaccines. Even if we ever got to the point where we could make vaccines, the likelihood that we would be able to ramp them up quickly enough to do a big part of the world's population was a pipe dream. At that point in the province of Alberta, we were on average immunizing 5 to 10% of the population for influenza, and that was taking us months. So at every level, we just weren't ready. And I just want to contrast that. Why I say that what you've done is impossible is that it's been less than a year since major public health action was taken to limit the spread of the virus in Canada. And in that time, ourselves and the world have determined the sequence, designed multiple vaccines, tested them and proved their effectiveness and safety to regulators, manufactured them in tens of millions of doses, and in some countries have significantly decreased deaths and transmission in a year. That's amazing. And just to give one other point, any given year, influenza vaccine has an efficacy of 30 to 70%. To have these things routinely testing at 80% or 90% is just unbelievable. So I'm going to go back through a couple of points for each of the presenters. Joanne mentioned that vaccine development never occurred at this place before. And she also pointed out how stellar the efficacy has been. I'd like to point out, though, that the one thing that I think people haven't commented on much is the new technologies are no longer dependent on the timelines of the time-honored ways of developing vaccines. And so I mentioned eggs, but that's true across the board, is that if you have to retool a messenger RNA vaccine to be able to handle a new variant, that's actually relatively simple in comparison to the processes we would have had in the past. Can't say enough about how impressed I am with our regulatory colleagues and how they met the challenge. It's a tough place to work because you can't afford to make mistakes, and everyone knows that doing stuff good, cheap, and fast, you can only do 2 of the 3, and the regulators have managed to actually do all 3 really well. I think the ability to use parallel instead of sequential processes is something that will stand us in good stead in the future, that rolling submissions are important. And I also think that the proactive consultation leads to a relationship which was never adversarial between vaccine manufacturers and the regulators, but certainly what's being described is a lot more collegial, and so that's a really good thing. I think that the transparency that they talked about is absolutely vital to recognize, and we now know that that transparency is important, not just so that everybody in the public can make informed decisions, but it's going to be a vital tool to be able to use to ensure that vaccine uptakes are what we need them to be. The one thing that you left out that I think is going to be very interesting for the future, particularly if you face a pandemic with significantly worse mortality, is the importance of working out when and where challenge trials can be done. And I think that will turn out to be one of the hidden gems, that the work that had to be done on the ethics to make sure that that was done correctly in a life-threatening situation for the planet will be very important. What can I say about NACI? If there's a group of people, a smaller group of people who works harder and puts a better quality product out on the planet, I don't know who they are. And one of the things I think will be key for them in the future, which they already have done very well, is that we're seeing a battle where evidence doesn't mean what we thought it meant, and we're going to have to go beyond that. As the technical experts, they've already shown sensitivity to that, but that's going to be a battleground in the future, making sure that the evidence we produce is persuasive to people. I mentioned that something that I am a little concerned about, which is that there's always a tension between evaluating the evidence and making recommendations and then seeing them be carried out. And you mentioned that the federal government doesn't have to accept the recommendations, and of course the provinces don't have it as binding. It's a measure of how great NACI is that is almost never a problem. The province sign on because they respect and trust the quality of the work. But we've seen some places where trust breaks down in this, particularly with governments making decisions and not being prepared to show their work in public. And so I think that's one that I'd be interested in hearing about in the future, is how NACI would not necessarily challenge a decision not to follow the recommendations, but be able to make people aware that they had made those recommendations. And then Jeff's work on vaccine surveillance in Canada is cutting edge. I think, as he correctly identified, he wasn't going to talk about vaccine confidence and program evaluation, but those are the 2 things that I really hope would improve in our ability to inform in the future. He talked about some targeted studies to take a look at things like inequity, and I can only emphasize that not only are they as important as he thinks they are, but we should be routinely measuring inequity gradients about everything we do in public health, routinely, so that we can do it in real time. It's embarrassing to me that it was academics who had to step in and in real time pull that information out. That should be a routine part of program evaluation across the board in public health. So good for the academics, but bad for us. So I just want to pull back and then give a couple of other things I'd like to recommend that may not have been as fully dealt with in the 4 presentations. There has been a continuing cycle going on in this country since at least the 1990s to use the influenza season to assess, plan, act, evaluate, and reflect on how we can move closer to a better ability to adjust to a pandemic influenza. And a lot of the progress that's been made has come out of that very humble, incremental, year-on-year cycle. And so I would recommend that whatever happens with COVID, that we learn from that and make sure that we do take time to reflect and then improve what we do on the next level. The second thing that's worked really well has been a distributed leadership, including things like program evaluation, and being able to check across the country to find out who's really solved an issue that is of concern to other people in the country, and then being able to use that evaluation to publicize that best practice and see how well we can get it adopted across the country. That might be a role for a pan-Canadian network, but it's one that I think would repay any kind of investment that was put into it. And then the last thing I'd like to make a plea for is that even though my background is a lab and in surveillance and epidemiology, there will be calls to put lots of money into databases and IT and computer programming, but I would actually recommend highly that whatever we do next, including in the 4 areas that were described, is that we need to invest in people. And there have been times when the federal government has invested hundreds of millions to billions of dollars in improving our ability to handle data and information. But if you translate that into what the effect of putting epidemiologists in every single province in sufficient numbers to improve what we do, you would automatically get pressure to improve our information system. So if you think about an epidemiologist that, let's say, an underpaid epidemiologist at $100K for a 30-year career, that's $3 million. And then if you were to invest $100 million, you'd have 33 of them to distribute across the country. And that would benefit the amount of work that we can do in this area for 30 years with them equitably distributed across the country. I can't imagine how much better off we'd be with that investment. So that's my last plea is I'm not trying to choke off the computer guys and the IT guys, but let's invest in the humans whose careers are going to last long enough to make a difference for the next pandemic.
Okay, well, thank you very much, Jim, for so thoughtfully pulling this all together. A couple of things. We are at the end of our time, so we're not going to have time for a question and answer. There were a lot of questions that were addressed through the speakers' comments that they made, and then some others, Jeff Kwong especially had a chance to respond to some of the questions here. Today's workshop, the recording of it will be available on the CanCOVID website shortly for people to review. And so I think we're going to pull it to an end there. I just want to thank all of our speakers, Drs. Langley, Bettle, Quach, Kwong, and Talbot for their great presentations to bring us right up to date on where things are at with COVID-19 vaccines. And I'll just echo Dr. Talbot's final point there, which is the, going forward, there's a lot of smart people in Canada working very hard in public health, infectious disease epidemiology, vaccine research, but the group is relatively thinly spread, and some enhancement of the workforce related to this is probably, hopefully, one of the lasting things that can come out of this in terms of a positive thing going forward. So with that, I'll just say thank you to everybody for who participated in listening today, and hope that you got out of it what you were looking for, and please stay tuned for announcements on the upcoming CanCOVID workshops and seminars and lecture series. So thanks everybody for participating today.